Thank you for permitting me to give this journal club. I'm sorry, with the COVID vaccine, I wasn't able to give it last week. So we're kind of already running in time, so I'll just jump into it. Disclosures are the same as last week or last time. Today, I am going to be discussing a paper that I actually published about folate hydrolase 1 expression in Merkel cell carcinoma and how this expression can be a target for antibody-based brachytherapy. We're going to go over a few different topics throughout the entire talk. First, we're going to go over history. So Dr. Neil Bander was recently named one of the founding fathers of PSMA technology. And in 2014, he offered me a postdoctoral fellowship position. And I had come into this position with no prior urology experience, but I had multiple years of postdoctoral work in dermatology. So I had quite a bit to learn for this new postdoc position. And Dr. Bander told me, okay, I had to review prostate anatomy, at least kind of get an idea of where it was. And what is prostate cancer? What is prostate-specific membrane antigen? And J591, which is the anti-PSMA antibody that he had invented. And so this is an actual picture of my apartment at that time. And you can see all of the manuscripts kind of on the floor. And I started reading literally every word of every manuscript ever published from Dr. Bander's lab. And so one of the first papers I read was from 1997. And, I was really confused. So I'm supposed to be learning about prostate cancer and prostate-specific membrane antigen on prostate cancer and how J591 is kind of a carrier molecule for this target on prostate cancer. But then I saw these extra things like lung and breast and liver, and I said, so wait, why are these things being mentioned? So I said, okay, all right, next paper, 1998. And this paper was like, okay, well, now I understand the mechanism. I understand this. Okay, so this is basically, we see J591 and it attaches to the membrane receptor right here. And, as soon as it attaches, that induces something in the cell to eat J591 and whatever was tagged with it or whatever it was carrying. And then it remains inside the cell for the remainder of, well, if it's a radioactive isotope, for the remainder of the decay of the isotope. But then, and I read another paper in 1999, and again, I faced that initial kind of confusion. So again, I saw, okay, we're talking about prostate-specific membrane antigen and targeting of prostate-specific membrane antigen, but we were targeting it in renal cell, bladder cancer, colon cancer, glioblastoma, pancreatic cancer, sarcomas, breast, vascular, testicular. And then I recognized something, melanoma. And so I barely knew where the prostate was, but I knew a lot about cutaneous oncology from my prior postdoctoral fellowships. And so I asked Dr. Bander if we could do some research on some skin cancers and use his antibody. And he told me that as long as it didn't interfere with J591 as used in prostate cancer, that I could expand, I guess, what we were doing. And so we established a collaboration with my colleague, Barbara, and her PI. I'll continue. So now I want to get back to that whole prostate-specific membrane antigen. That was very confusing for me when I started working for Dr. Bander. Why is it called prostate-specific membrane antigen? And I started reading more about it, and it seemed that they had done some expression, protein expression profiles, and also RNA expression or gene expression. And So based on the protein expression, which was determined with antibody staining, yes, you could see that the prostate expressed strong or +4 level of antibody staining, but so did a bunch of other tissues. I mean, the duodenum, the small intestines, and the kidneys also expressed moderate or a lot. I mean, it's +3 PSMA expression. So again, we're saying it's prostate specific membrane antigen that happens to be on other tissues as well. And so what is prostate specific membrane antigen? Well, it's a transmembrane receptor. And this receptor, what we know is that in the context of the duodenum, it can cleave terminal glutamates and help with folate absorption. And in the brain, it can cleave terminal glutamates and make the neurotransmitter glutamate. And when something binds to this receptor, it somehow induces the cell to endocytose that receptor. So something binds to the receptor, and then through this molecule here, it interacts with these two proteins, and that's what induces the internalization of a carrier molecule and its payload. So where is this expressed and when is it expressed? So physiologically, prostate-specific membrane antigen is expressed in the prostate and a bunch of other tissues, as we previously saw. And functionally, in this physiological expression, it helps with folate uptake in the small bowel, particularly the duodenum, and in glutamate production in the brain, as I mentioned before. And pathologically, PSMA is expressed in prostate cancer cell, but it's also expressed in the neovessels of virtually every single type of solid tumor. So the functionality of pathological expression of PSMA in these tissues, we don't really know. We don't really know why tumors upregulate PSMA. And another important point is that in the physiological expressions of PSMA, these expression patterns aren't as easily exposed to circulating antibodies or small peptides due to tight junctions throughout the body. So this was the first project that Dr. Bander put me on. And this is what I think is a very important, because in this paper, so in this study, what we did is we took the supernatant of multiple solid tumors, different solid tumors, and then we took HUVEC cells, which are undifferentiated endothelial cells, and we were able to induce the expression of PSMA in these neovessels associated with these different tumors. We also identified a protein that was secreted, I guess, by the tumor cells, and it was found in that supernatant that caused the upregulation of PSMA in the HUVEC cells. And this is, to my knowledge at least, the only mouse model that exists for PSMA expression in solid tumor neovessels, because we know that mouse neovessels don't express PSMA. And so I finished that project, and I was like, that name, prostate-specific membrane antigen, it really irks me. It really bothers me because it's not so prostate-specific, right? So here we have a PSMA PET-CT, and you can see that there's pretty high metabolic activity, or high not metabolic activity, but high uptake of the positron emitting folate 1 targeting molecule in the duodenum, and because it's expressed in the duodenum, and in the duodenum it's called folate hydrolase 1, which also happens to be the name of the gene that encodes this expression. And so, at this point I had two important conclusions. So number one is prostate-specific membrane antigen isn't so prostate-specific. And then number two is, we should really call it folate hydrolase 1, which is, in line with the name of the gene that encodes it. And so I published just that in the Radiation Oncology Red Journal. And I proposed the argument for, establishing a more correct nomenclature for this antibody or for this target. All right, so before we get to the actual paper, I wanted to review a few basic concepts of therapeutic radiation. So why am I reviewing this? Well, nuclear medicine started off as being a therapeutic kind of field, and then we kind of swung the pendulum over to mostly imaging based, and now that pendulum seems to be moving back towards therapeutic with the recent FDA approval of Pluvicto and Xofigo and these these therapeutic agents. So when we think about therapeutic ionizing radiation, we want to think about, okay, well, is it charged? Is it uncharged? Is it causing a direct effect, or is it causing an indirect effect? And so when we talk about photons, we're talking about uncharged ionizing radiation. And I like to use the analogy of a prodrug, meaning photons aren't active from the moment they enter a cell. In fact, a photon can actually go through an entire human body and never interact with any tissue at all. And photons really only become active when they transition from being a photon to an electron in tissue by interacting with the atoms in the tissue. And then photons will kind of continue moving out in tissue, and they'll slowly lose energy in a few events that are generating these electrons, or they'll just pass through the body. Then the active drug, or electrons, which would be, in our case, beta emission. Electrons are active from the moment they enter tissue, and they're capable of DNA damage immediately. They interact with every atom they pass by, and they gradually lose kinetic energy as they move through the tissue and slowly and interact with all the molecules. So now I went back to my radiobiology text. It's the 7th edition from when I was in residency. There's a newer version available. I'm not sure how changed it is, but the first thing we need to think about is what is direct and indirect action of radiation. And so the magic number that I guess we were taught was that with ionizing radiation, about 1/3 of that radiation is direct, which means that it is the actual electron, either by beta emission or the electrons that are generated from the gamma, so from the photons, that it kicks off an electron in some atom in the tissue that can interact with water and create reactive oxygen species, hydroxyl radicals, that then diffuse a short distance and nick the DNA. Now we're going to define 3 important terms. The first one is oxygen, the oxygen enhancement ratio. So oxygen does have an effect on the survival of this tissue, and it really acts at the level of the free radicals, right? So reactive oxygen species, a hydroxyl radical, they all have oxygen in them, right? So we're enhancing the indirect effect of radiation. And so here, so the definition of oxygen enhancement ratio is the ratio of doses administered under hypoxic to aerated conditions needed to achieve the same biological effect. And over here you see survival curve. So you see that here in blue, in the hypoxic conditions, the cells are dying, but they're doing pretty well. And then as soon as we add oxygen and increase that secondary or that indirect effect of ionizing radiation, all of a sudden the cells aren't doing as well. They're not surviving as much, right? We have a much steeper slope downwards. Okay. So this, I think, is a phenomenal figure. I really want you to take a look at it while I let you know what linear energy transfer is. So linear energy transfer is defined as the energy transferred per unit length in micrometers of the track. And the unit for this is kilo electron volt per micrometer of unit density material. And so I think this figure really allows you to see what linear energy transfer is. So here above we have protons. And so when we look at low energy protons, like 500 kilo electron volts, we see almost like this thick line of ionizing events going straight across, the organelles in this cell. But then interestingly, when you increase the energy of protons, which are, large charged particles, we see a more sparsely ionizing pattern, right? And this kind of looks like this down here, which is 5 kilo electron volts. So fast protons kind of ionize at the same rate of 5 kilo electron volt electrons. And so this is linear energy transfer. It's how much ionization or energy is transferred to the tissue on the track of that particle. And here you can see it a little bit bigger. I just wanted to put this picture in here now because this foreshadows what I'm going to show later on. But here in this picture, we are on a different scale. And so we're in 50, so in 1 micron, in this scale that's to 1 micron, we're seeing the actual organelles of a cell. But if we zoom out a little bit, we can see this over a larger area and even maybe cover an entire tumor. And then so here we can see all these circles here are nuclei of cells. So this is, so we're seeing a bunch of cells here. And we can also superimpose these ionization events from the linear energy transfer onto this image. Okay, so what is the relationship between the oxygen enhancement ratio and linear energy transfer? So it's very simple. Basically, oxygen really helps sparsely ionizing radiations like X-rays because it increases that indirect ionization effect, but it doesn't have that much of an effect on, larger high LET particles like, alpha particles or protons because they have a more direct component of damage. And the last thing I wanted to cover is absorbed dose. Absorbed dose is measured by the unit gray, and 1 gray is equivalent to 1 joule per kilogram, and that is a volume of tissue, or well, in this context of tissue. And another last important point is that equal doses of different types of radiation do not produce equal biological effects. And I think that's because of the proportion of, direct and indirect damage that each of the different radiation types cause. And, the present or the lack of oxygen and other factors that go into play. All right, mechanism of action. How does radiation actually produce their clinical effect that we want to see? And I put in this, I cited this paper I published in the British Journal of Dermatology, and it seems kind of like out of place, but and it's about keloids. Like why is it here? But the reason I put it in here is because I really describe the mechanism in the context, in a different context of keloids, but it describes kind of how ionizing radiation produces the therapeutic or clinical effect that we want, and it's by two different forms of cell death. So whether it's indirect or direct damage to the DNA, eventually that DNA damage could result in a type of death called either interface death, which is what we're going to review here. And interface death is a common type of cell death that we've all learned about many, many times over and over again throughout our education, and that's apoptosis, right? So that is, the damage is so bad, the cell cannot divide again, and it just, triggers suicide. It decides, all right, that's it. It's over for me. And with apoptosis, you'll see membrane blebbing, fragmentation, and eventually the cells around it will eat it and take its useful proteins and whatnot inside of the cell. All right. So the other type of death which is actually the more prominent form of death with ionizing radiation, is called a mitotic death. And the reason we know this is a more prominent form is because, when we radiate tissue, we don't see a bunch of holes in the tissue, right? We see like tumors can actually stay there, the prostate, right? External beam irradiation of the prostate, people that have received that treatment, they still have their prostates, right? But why isn't it cancerous anymore? So it's because of mitotic death. So mitotic death is a form of sterilization or fixation. Why? Because the cell remains physically present. It's morphologically, it's intact. And it's able to survive. It's able to stay alive. It's able to make proteins and synthesize RNA, but it can't divide anymore. It can't proliferate, which is fine, because the problem with cancer is that it's breaking all the rules and it keeps dividing. So if we could just stop the division, that's good enough. We got our therapeutic, our clinical endpoint. And I just want to mention the mean lethal dose for sterilization and fixation is actually 2 gray. So that's not a very high dose. Okay, so delivery. How do we get radiation, ionizing radiation, into tissues? All right, so we have multiple modalities. So for electrons, we can use linear accelerators or brachytherapy in the form of beta emitting isotopes. Or for photons, we can use orthovoltage devices. We can use linear accelerators or brachytherapy using gamma emitting isotopes. And so I want to make a further distinction that for brachytherapy, we can use sealed or unsealed sources for this. So sealed sources really relates to like high dose rate brachytherapy or low dose rate brachytherapy, and unsealed sources usually relate to isotopes that are used in radiopharmaceutical targeting, like Pluvicto. So what is brachytherapy? Brachytherapy is a form of radiotherapy. It actually comes from the Greek root brachy, which is short, and it means to bring the radiation source directly right close to our target cell or target tissue, whatever it is. And we can accomplish this type of radiotherapy using catheters, needles, metal seeds, antibody conjugates, or small peptide conjugates. Okay. All right, so now I'm going to create a bridge. I'm going to bridge catheter-based brachytherapy with a case, and I'm going to bridge it to molecule-based brachytherapy, which is kind of a carrier-based brachytherapy. So using an antibody or a small molecule to carry a isotope payload to a target cell. Okay, so this is a catheter. And we're gonna, for this presentation, we're just gonna talk about catheter-based with gamma emission. Gamma emission is photon emission. And I'm gonna bridge that to molecule-based brachytherapy. Here we go. And this is really talking about beta emission, because we're using lutetium, and that's electron emission, okay? All right, so the first step in catheter-based brachytherapy is you put the catheter into the tissue that you want to treat. And then you do a CT simulation, right? So you get a CT with slices of that patient, and you can see the catheter. And then that CT simulation is used to superimpose a dose distribution that's calculated by some sort of Monte Carlo simulation, right? And so we kind of do the same thing for our Pluvicto treatments, right? So patients undergo a PSMA PET CT, and you know, while we don't really do the superimposing of dose distribution or any formal dose calculation at target sites, we're at least able to see whether there are tumor deposits and if those are suitable for targeting with this, small molecular or antibody based brachytherapy. And here I don't have any PSMA PET CTs of solid tumors available, and I'm sorry for that, but I do have an indium 111, and this is the one from a prior presentation of mine, and this is metastatic melanoma. So in a patient with a non-prostatic metastatic solid tumor, we can identify these are all potential sites that we could treat with this treatment. All right, so the treatment planning process. So how do you do it for catheters? So each of these points along here are called dwell points, okay? And so we need to prescribe 6 gray per fraction. And so by calculating the dose distribution, we can do kind of an inverse treatment planning. We could say, okay, so how long does the sealed source have to stay at each of these dwell points, okay? So that the dose distribution in the region that we want to treat equates to 6 gray. And so the software will calculate it for us. Oh, I don't know, I did that. We'll calculate it for us. And in this particular case, it says that, in the dwell position, so the isotope would have to go here and stay for 6 seconds, then 7.6, and 7.6, and so forth. And at the end of this stay, it'll retract, and we have effectively delivered 6 gray. Okay, and so for molecular-based brachytherapy, radiation treatment planning isn't done. At least not yet. Okay, and now the treatment part. So again, like I mentioned, this is kind of like the little home, this Wally looking thing is the home for iridium 192. This is the tip of the catheter. This has the this is where the sealed isotope is, so where the iridium 192 is, and this will travel along this catheter, and it'll stop at the dwell points for the defined amount of times, and it'll slowly irradiate along that length, and at the end of it, we have delivered 6 gray. For Pluvicto treatment, it's a little bit different because, the mechanism is, we know that it reaches a cell surface and then it's internalized, and effectively, because it's internalized and captured with the cell becomes the dwell point for the remainder of the radioactive decay. All right, so now we've reached my my paper. Okay. All right, so Merkel cell. Merkel cell carcinoma is a relatively rare cancer, but it's pretty aggressive. It's actually been characterized as being 3 times more lethal than melanoma. And we already think melanoma, and we think that that's a pretty aggressive cancer. So, imagining a cancer that's 3 times more lethal is a little scary. And Merkel cell is associated with a very low survival rates, especially when patients have developed metastatic disease. And unfortunately, 30 to 50% of patients with locally staged disease will eventually develop distant metastases. There was a phase 2 trial of an anti-PD-L1 monoclonal antibody, and this is checkpoint inhibition. Basically, this type of therapy, what it does is it causes the immune system to recognize the cancer cells a little bit better. And this type of therapy actually resulted in a 32% durable response, and that resulted in FDA approval after a phase 2 trial in March of 2017. Unfortunately, while that's great, 50% of the patients don't respond to this type of therapy for Merkel cell. And importantly, is this unique opportunity related to Merkel cell carcinoma, where because it's a rare disease, the FDA actually provides assistance, financial and logistical incentives to find therapies, potential effective therapies that can help these patients and improve survival, particularly in these patients that don't really respond to PD-1 and PD-L1 axis targeting. So radiation in Merkel cell, theoretically, because Merkel cell carcinoma has a large mitotic fraction, that means it has several cells that would be theoretically very sensitive to ionizing radiation damage, right? That's one of the times when the cell is most sensitive to radiation. And Merkel cell carcinomas are also highly vascularized, so that means they have a large blood component, a large blood flow, which brings oxygen, right? And so what we learned before is that oxygen can enhance the indirect component of sparsely ionizing radiations like photons and electrons. And currently, external beam radiation is recommended for the following indications, when surgery is impossible, when margins are positive or borderline, and presence of lymphovascular invasion, and if the patient is immunocompromised for any reason. All right, so what did we try to do? So we thought, okay, what if we can target disseminated metastatic Merkel cell carcinoma? And sorry about that. What if we can target disseminated Merkel cell carcinoma? And so we collected this very rare tumor from multiple academic medical centers around the world. So from Switzerland, Czech Republic, Germany, and a couple of sites in the US. And I just want to draw your attention to this little arrow and that little spot right there. So that's Puerto Rico. That's my home. Okay, so all right. So after collecting all these samples, we were able to collect 81 Merkel cell carcinoma tumors. And this is a very large number. I don't know if you remember the last presentation I gave, but there were only, I think, about like 40 melanoma cases that were reviewed, and they made all these calculations based on that. So this is a very, very large cohort for a rare disease. And then we performed immunohistochemistry to evaluate the expression. The primary antibodies we used were these. 3E6 targets folate 1 or PSMA. Then we use this mouse IgG1 isotype. That's just like just an antibody. It's not meant to really bind to anything. And then we also used anti-CD31. And so our negative control was this mouse IgG1, and that was non-specific. It's just supposed to it's just supposed to kind of say, like, let you know, okay, there's not random non-specific binding just because an antibody is present. And then this was our positive control was anti-CD31, which binds to CD31, and CD31 is a marker, a very specific marker for blood vessels and endothelial cells. So all endothelial cells do express this target. And then the secondary antibody we used to give us our our staining was this one. Okay, so here's an image of our this is the tumor, and this is higher magnification of the tumor, and you can see that there's no staining visible with the isotype control. All right, so positive control, we can see that when we use the CD31 for blood vessels, we get all of this staining. So all of these are blood vessels within a tumor. And then when we stain for folate 1, it's kind of hard to tell in this, this magnification, but when we go a little bit closer, so these are blood vessels. These are blood vessels, and they have some staining for folate hydrolase 1. All right, so of our 81 cases, 67% of them expressed folate 1. Primary cases, 77% expression, metastatic cases, 60% expression. And what we saw over and over again is that the neovessels, folate 1 was restricted to the neovessels. There was no staining of the normal vessels in the periphery of the tumors. All right, so after we stained all of these sample tumors with immunohistochemistry, we wanted to quantify the amount of staining. And so we use this kind of a standard semi-quantification, which you saw previously in the prostate gene or PSMA gene expression profile. So negative was, no staining. Then we did +1, +2, +3, and +4. +4 is kind of maximal staining, and that's what's more seen in prostate cancer, where you achieve staining of the cells and the blood vessels. And then these so once we had all of our everything stained, we gave all the slides to 3 dermatopathologists that independently reviewed and scored all of the slides. And fortunately, there was no discrepancy between the dermatopathologist. But if there had been, we would have taken the score that was that had the majority vote. And so what we found is that Merkel cell carcinoma expresses significantly more folate hydrolase 1 as compared to healthy skin in both primary and metastatic disease. And when we look at the distribution, we had quite a bit of +3 staining, so that would indicate that there was more target at the site, and also quite a bit of +1 staining, which means there is a target, at least. Okay. All right, then we did immunofluorescent co-labeling. And so the point of doing this is that we show that a blood vessel, this is the staining for folate hydrolase 1. And then that same blood vessel, if we use if the primary antibody is switched to an anti-CD31, which is specific for blood vessels, we see the same staining pattern. Right? And then if we merge the two and, kind of turn on the DAPI so we can see the nuclei of all of the surrounding cells, yes, we confirm that folate 1 expression is restricted to these carcinoma neovessels. Yeah. All right. Then what we did is, and this is a part that kind of came later, and was actually inspired by the situation when my mother developed metastatic disease, and I said, we what if standard of care doesn't work? We need to figure out how to calculate a curative intent dose, and then I said, okay, all right, so can we do a Monte Carlo simulation of a point on a vessel, and just see what the simulated ionization or dose distribution would be. And so that's what we did. So we just did a simple simulation. We did the emission tracks of 30 electrons of lutetium. We chose the mean energy of 0.147 million electron volts, irradiating from that point in one direction on one two-dimensional plane, but really it was spherically in the it irradiates spherically, and then once we did that, we superimposed this simulated distribution of the 30 electron emissions from the lutetium and in water onto the immunofluorescent histopathology. And that's kind of the same as what we do in, radiation oncology or catheter-based brachytherapy, where I went over that we do the CT simulation, which is a non-contrast CT, and then we superimpose the Monte Carlo simulation of dose distribution on top of that CT, and that's how we determine the calculated absorbed dose in that tissue. So this is very similar to that. And also, I should mention that this looks like a bunch of little dots versus tracks because electrons don't really follow that straight line like the protons kind of do. They kind of move in and out of the field, so you're just seeing the electrons as they're kind of going in and out of the plane. All right, so another really important thing to kind of take home from this image is the fact that, remember I said this was DAPI, and these are cell nuclei. So around this is our full cells. But if you look at the ionization points, you could imagine that any point, any of these yellow kind of spots that overlap the blue DAPI, that's a potential for direct radiation damage. And direct damage, can result in basically that interface or mitotic cell death we spoke about before. So that's like apoptosis or just self-fixation sterilization. All right. And okay, so this is just kind of illustrating the point again. It's very similar to this, right? So this would be the electrons or the ionizations from a proton, and they're in a straight line, versus, the electrons, they kind of scatter a lot more because they're small, right? They kind of get knocked around versus a proton will just power through. Okay, so now I'm gonna introduce, in 10 minutes, I'm gonna introduce microdosimetry. All right, so I'm gonna have to move a little quickly. So this is, again, brachytherapy, catheter-based. We recognize some figures from before, Wally and the catheter, the dwell points in the ear of that patient. And again, the dose, it starts off, it's sequential, it's step-based. So you're at a certain dwell point for a certain amount of time to release a certain amount of energy that can be calculated. Oh, wait. Okay, there we go. And so based on this information, with the dosimetry software that we have right now, whether we're using electrons or it's, that high dose rate brachytherapy, we can simulate the dose distribution based on the characteristics of the isotope being used and the location of the source, or the source of the electrons or photons. Okay, so what about, so I've already kind of gone over this catheter based brachytherapy, and it's really pertinent for high dose rate, but I haven't actually discussed anything about low dose rate brachytherapy. So I'm going to touch upon that because that's really important to see how we're going about the microdosimetry. Okay, so this is a classic form of low dose rate brachytherapy, which is used for prostate brachytherapy. So here, this is a rectal ultrasound, and that creates this image of the prostate, which can be contoured. And so here's the prostate being contoured at different depths going in a stepwise fashion of the ultrasound. And then needles are put into the prostate, and they leave seeds, radioactive seeds in the prostate, and those seeds act as that point source, or that dwell point. However, it's stationary. They stay in there, they don't come out, they don't move, and they basically emit, you calculate the radiation dose from there as radiating spherically around the seed, and it decays until, or the dose is deposited until the full decay of the isotope. Okay, so here is the radiation treatment planning for prostate brachytherapy. You kind of expect a uniform kind of distribution of the needle insertion points and how many seeds go into each needle. You see that there's two seeds going into here in the center because the urethra is there, and you really don't want to heat up the urethra too much, because that'll cause problems. And then in the periphery, you want to give as high a dose as possible because you're away from the urethra, and you just want to like make the prostate really, really hot. And then when you insert the parameters of whatever isotope you're using, whether it's I-125 or palladium-103, into a Monte Carlo simulator, you can actually calculate the anticipated dose distribution and what the delivered dose would be to the prostate. All right, so microdosimetry. Okay, so again, we know we can input the parameters of iodine 125 or palladium 103 into a simulation software, and we can calculate a dose for a specific volume of tissue, which in this case is a prostate, right? But let's just forget it's a prostate. We could do the same thing. Well, what if we just inputted all the parameters of lutetium 177 into a Monte Carlo simulation and calculate a dose? Okay, yeah, it's possible. There's no reason we can't do it. And we know I did it just a few slides above, right? Remember the histopathology and the immunofluorescence and the superimposed ionization points from emission 30 electron emissions of lutetium, right? So the point is we can do it. But to do microdosimetry, so I created this kind of very very simplified kind of workflow overview of how to perform it. Okay, so you need to have a little bit more information than just the parameters of lutetium, right? So we need to know how the dose is going to be distributed within this specific volume of tissue that happens to be a lot smaller, at least for initial calculations, will be a lot smaller than a prostate, right? And we have to identify where the location of these sources, these point sources are, right? So we could kind of imagine that, and if you look at these are the prostate seeds inside of the prostate, right? And this whole thing here is a prostate. But like if you look at the Merkel cell carcinoma tumor, you also get all these like spots here, and it's kind of like the seeds in a prostate, right? So if you assign to each of these spots, right, as a point source, and you can input the lutetium parameters, and you can potentially calculate a dose distribution. But to do that, we need to know how much radiation will be in each of these points, right? And so to do that, we're going to have to know the average number of receptors that are going to be there. Okay, and so in the prior paper, I showed that we can at least semi-quantitatively average the amount of receptors in a certain area. Okay, it's not perfect, but I think it would give a good starting point to work from and to build upon and to become a little bit more specific in the future. Okay, then we need to determine what is the total length of that these receptors will be on, that will be emitting the isotope, kind of like the same thing as the catheter, right? You have a specific length and the dwell points along each length, right? So we need to do that. And so to do that, we can draw upon what we already know from catheter-based high dose rate brachytherapy. And so to kind of bring it to scale, so we can use a unit length of, for example, 1,000 micrometers, which is equivalent to 1 millimeter, which, we can easily do a CT of 1 millimeter slices, right? And then we can assume that each slice of the CT would be a dwell point of 1,000 micrometer thickness. And in that 1,000 micrometer thickness, we'll have a certain number of folate-1 molecules. Okay, so other information that we need to also include is, okay, so what's the average total length that's going to be treated for a given tumor size? And so I couldn't find a picture online, and this, I'm sorry, this was very late last night, but so we can make assumptions. We can say, okay, well, we'll distribute it. So these are all the spots where the neovessels are, and then we can just estimate, average it out, and assume that they just go straight through the whole tumor size. Okay, so that's one way to determine a length that we can input into the Monte Carlo simulation. Okay, so then we have to make a distribution assumption, and that we can just draw upon, like I've already kind of done a few times, upon what we know from metal seeds, low dose rate brachytherapy, right? So the similarities there. And we can draw upon that and apply them to neovessels that stain positively or that express folate1. Okay, then we need to determine the number of lutetium molecules per lutetium atoms per carrier molecule, and that's going to be specific to the drug. For J591, there's about 4 atoms per antibody. I'm not sure how many lutetium molecules are on the Pluvicto carrier. Okay, the last thing we need to gather, which we already have, is the lutetium characteristics, the mean energy, the range of this of the beta emission, and we input all of that into the Monte Carlo simulation software, which also already exists. And this particular software is catered specifically to brachytherapy, and it was actually developed by the physics department at McGill University. And so we gathered all of that information. Once we gather all of that information, or are able to gather all of that information, we input it into the Monte Carlo simulation, and that should give us an estimated absorbed dose. And that is work to be done in the future. So this is a potential mechanism to be able to do radiation treatment planning for radiopharmaceutical targeting of folate hydrolase 1, or also PSMA. So the conclusions here are folate hydrolase 1 expression within Merkel cell carcinoma associated neovessels is prevalent, and this expression is significantly more in the neovessels as compared to healthy skin or healthy vessels. And also targeting of the neovessels results in highly localized dose distribution because of that range of the lutetium to I think it was 2.2 millimeters. Don't quote me on that. And also with this introduction to microdosimetry, it seems that radiation treatment planning might really be possible in the future. And if it becomes possible, then it's definitely something that we should develop towards incorporating into our clinical practice as nuclear medicine physicians. All right, so thank you, and it's exactly 9 o'clock. So are there any questions?
Thank you very much, Dr. Ramirez-Ford. Just one quick question. Do you have any clinical case where you have a gallium PSMA scan in a Merkel cell tumor in a clinical setting?
No. So
is it super hot? I don't recall personally seeing one, so I'm sure there are case references in the literature, but I haven't looked at it. But I'm just checking. Have you ever looked at that?
Oh, you mean
how intense the uptake is of the PSMA within the Merkel cell?
PSMA in Merkel cell carcinoma? I've never seen one. I don't think anyone has done a folate 1 PET CT on a Merkel cell carcinoma patient yet. At least at Cornell, we had a solid tumor trial, and some of this work has already been published. And we were able to use, PSMA PET CT or folate 1 PET CT and show sites of metastatic disease in a whole range of different metastatic cancers, like pulmonary adenocarcinomas, melanomas. I think we published 4 cases on that.
But that was all with indium J591. Is that correct?
Oh, no. It was done with zirconium 89. I just don't have the images. But I could potentially ask Dr. Vander to see if he has any images. Before I made the last presentation, I had asked around the department to see if they had any solid tumor cases, but there weren't any, at least in our department. So, but other departments have done this staining or this PET CT in other tumor types, and there have been several reports published in the Journal of Clinical Nuclear Medicine, and they show that it's pretty avid uptake in melanoma metastases. So you can presume that based on the staining profile of the tumors, of the Merkel cell tumors, you would expect the same thing. It's already been shown in so many other solid tumor metastases that I wouldn't anticipate that PSMA PET CT or folate 1 PET CT of a Merkel cell carcinoma tumor wouldn't express or wouldn't show avid uptake of the tracer. It's just a matter of having a patient, not having any more options, and finding a nuclear medicine physician willing enough to push this recommendation forward in the tumor board.
Right. No, I agree.